Yeah, subs aren't real teachers, right? No. They're just people that show up and collect easy cash, right? They're like, I don't care what happens with y'all, I'm gonna get paid either way, so.
Thank you for the worksheet.
They're not all like that. Subs who used to be teachers are trying to be teachers. I don't know who this guy is, I have no idea. Alright, ready? First, I need everybody in their seats. Don't worry about extra copies right now, guys. I need to take roll, and then we're going to show you Lucas's video. Hey, you posted it on, Lucas, you posted it on Schoology, right? Okay, I have it one more of the other. Go, dude. We're going to show 2 projects today. I also completely forgot. Yeah, how did you do? I need to get hers as well.
It's so good. Yeah, like, you get it on. No, it's just ukulele.
Oh, I know what you did. You sent it to me in a regular email, right? Photosynthesis rap, extra credit, hilarious.
Once through a little pretty.
Hold on, let's start.
Get your Snapchat out! Lucas, it's so good.
Guys,
He typed in hip hop. Oh my god.
don't laugh because you can't hear the lyrics.
It's so good.
Alright, this is, you have to have a rap name, right? DJ Joss the Blast? Whatever, I don't know what
Lucas Schmurda. DJ
Yeah. DJ Chlorophyll in the house.
Once through a little pretty leaf and needed some food or else it'd beaten some grief. Homie Sunlight, he wanted to join. He said grab some water and let's make some noise. But hold up, we're forgetting my bro. Stomata, carbon in the 2 fat O's. Yeah, we're getting glucose though. Chlorophyll stunting like the new fresh clothes. Then to throw it up in the air. Photosynthesis, just like you don't care. Even though you got the food that I need to eat. Can't rap like the Leaf or Mr. B. Era of the ATP era. Chlorophyll is back and ain't even scared of you, and the process is done. Photosynthesis, man, is #1. Photosynthesis, photosynthesis, photosynthesis. Yeah, job, Mr. Vera. Curious Sex.
Amazing! I will use that forever to show people exactly what I'm looking for. It's like 1,000 points extra credit is what that is.
Hey, I don't care. Like he's got mad skills. Like Sandra's mom. It was I'm excited.
It's also 1,000 points extra credit. Is the volume on? Yeah. Is your parents in the business at all?
What is this?
The headaches began on the 7th day after exposure. At first, it was just a pain behind the eyes, but then it grew worse. I decided to stay home from work and went to bed in my bungalow. The pain seemed to circle around inside my head. My personality changed. I became sullen, resentful, angry. My memory seemed to have blown away, but I was still not delirious. Not even aspirin could make the pain go away. 3 days later, I spiked a fever and began to vomit. The vomiting became extreme and turned into dry heaves. At the same time, I became strangely passive. My face lost all appearance of life and set itself into an expressionless mask.
Looks like my students during a lecture.
When I failed to show up for work, my colleagues began to wonder about me. After one look, they knew I needed to go to the hospital. It was too late for any help. The virus had already coursed into my bloodstream. The doctors examined me and could not come up with an explanation for what had happened. I needed professional help. There was a private hospital in Kisumu. They put me on a taxi to the airport, where I waited to board Kenyan Airways flight. When waiting to get onto the plane, I became extremely ill. I hunched over my seat with a bag over my mouth. I coughed a deep cough and regurgitated something into the bag. The bag swells up. It fills to the brim with vomit. The virus entered its final stages and finally took over my body.
Oh, let's see, both of them.
Yeah, I'll take that. You know what I saw? And then of course Whitney. What else would you? I like how the credits are like 20% of the length of the video. I was like, there's like another minute left.
Have you watched the credits?
I've seen like, yeah, left classics. Did you do editing, Jason?
Yeah, that's not good, Jason.
I thought they said Russell Crowe for a minute. I was like, wow. Impressive. Literally longer than the actual video itself. I think I stopped at this point in the credits. Okay, a couple questions, because I have 142 views already.
Yeah.
because I did forward it to the entire science department. So let's try and get you viral. Get it? Viral? Wow. Wow, guys. Viral? We'll get it viral. Okay, a couple questions. All of that footage was stock footage from other sources, or did you actually create?
Most of it I stole.
Okay, I would affect, like the ambulance, for instance.
Yeah, stolen.
Okay, obviously the plane stuff and the time lapse city stuff. The doctor scene where it scrolls up and shows all the videos, is that stolen too?
Yeah.
Okay, because I was like, is that his dad? Is it just happened to be at an MRI machine? Or like... Oh, okay. Where did you do the editing? At your house? Or...
No, I did the editing.
Alright, I expected to see some people running around throwing up all over the place. And then I hit play and I'm like, get my popcorn out. Really good. Far outdid my expectations of what I expected. So that's awesome. Yeah, I sent that out to the science department. Everybody's very impressed. As they should be. So I will be probably showing that video for the rest of my career, I would imagine.
It's a pretty short career.
It's gonna be hard to top. Really short? For the rest of my career. I don't even know what that means. Oh, like I'm gonna die or I'm gonna get fired? Damn, what's wrong with you guys? Horrible. Okay, I believe we left off.
Is that the tower?
Here. Did I show you any videos on glycolysis and Krebs cycle? Okay, we're probably right around here. Even regardless of where we're at, guys, I just want to review cellular respiration really quickly, so we're going to fly through this. The test next class is on photosynthesis, cellular respiration. It's not today. Not today. If you look at Schoology, it doesn't say it's today.
It said it was Friday.
Yeah, it says Friday. Which today is Wednesday. So next class, Friday.
Our test ever on
All right. Cellular respiration is where we take the food that we just made in the first process, photosynthesis, and now we're going to release that energy and put it into ATP. So photosynthesis, we took energy from the sun and we stored it into food, glucose. Now we're going to eat that glucose, breathe in oxygen gas, and we're going to release that energy and put it into ATP. The first part before aerobic respiration, before we go into the mitochondria and go through cellular respiration, we go through this process called glycolysis. All organisms go through this process. It's anaerobic, meaning it what? Anaerobic, this is. Does not require oxygen. Here's some things you should have in the margin of your notes. A, how many ATP are produced by this? How many ATP total are produced here? Net. 2. Remember, we spend $2 to make what? 4. We invest $2, but we end up making 4 of a profit of how many ATP? 2. So question number 1 that'll be on the test, know how many net gain ATP. Question 2, which this is what the stuff that you put on the margin, right, of your Cornell notes. Where is this occurring? Look at my picture over there. Where is it occurring? Is it occurring inside of the mitochondria or outside of the mitochondria? Glycolysis, the splitting of sugar. That's literally what glycolysis means. Glyco meaning glucose, lysis meaning separate or split. This occurs in the cytoplasm. Everything else, after we split, if we have oxygen, will occur inside of the mitochondria. So number 1, how much ATP? Number 2, where does it happen? Number 3, does this require oxygen? Yes or no? No. And if it does not require oxygen, what word does it?
Anaerobic.
Anaerobic. Anaerobic, exactly. So that's it. Those are the questions ultimately. And it happens first in this whole process.
Okay.
Okay? So real quick, about a minute, 20 seconds.
Glycolysis, which begins the breakdown of glucose, is a series of 10 enzyme
Cytoplasm,
catalyzed
outside of the
chemical
mitochondria.
reactions
inside of
can
a
be
cell.
divided into two main phases. In the energy investment phase, some ATP energy is used to start the process of glucose oxidation. By the end of this phase, a 6-carbon molecule, glucose, has been split into two 3-carbon molecules of glyceraldehyde phosphate. The 3-carbon glyceraldehyde phosphate molecules now enter the energy payoff phase. Chemical bonds are broken, and NAD+ picks up electrons and hydrogen ions, forming NADH. The energy released is used to attach phosphate groups. The phosphates are transferred to ADP, finally making some ATP. This way of making ATP is called substrate-level phosphorylation. A couple more reactions rearrange the atoms in the 3-carbon molecules. More ATP is generated in the final reaction that yields pyruvate. For each glucose molecule broken down during glycolysis, a net of 2 ATPs are formed, along with 2 NADH molecules.
Okay. So at the end of the day, we end up with pyruvate, a couple ATP, and a couple of those electron carrier molecules. Because remember, guys, photosynthesis, we ended up punking these two molecules right here. What are these two molecules?
Water.
Water, two molecules of water. Remember, we stole something from these guys. What did we steal from them?
Hydrogen.
What did we rob from these guys? We split them and took their what? Their what? Electrons. Because if you remember from photosynthesis, chlorophyll is the pigment that gets heated up by the sun. It absorbs the energy from sunlight. It absorbs it so much that electrons fly off of chlorophyll, which is a molecule made up of atoms, moves down the electron transport chain, pumping hydrogen ions into the thylakoid space. Now those electrons need to be replaced, and we get them from these two molecules. So we pop off these two atoms, which is hydrogen. This is one proton and one electron. We're going to steal the electrons from these two atoms, leaving them to be converted into a proton by themselves, which gets pumped into the thylakoid membrane. I'm going to take some electrons from that guy. I'm going to take some electrons from this guy. Water just got pumped, just got stolen, hijacked. I'm going to take your electrons. It got oxidized, where it loses electrons. What are these two atoms that are left behind? Oxygen. They bond together covalently, right? And form what molecule? Oxygen gas. This is what happens in photosynthesis. This is how plants release oxygen gas from water. Okay? Well, guess what? Oxygen. He's coming back for vengeance. I want my electrons back. So we breathe in oxygen. It goes all the way through our blood from our lungs. Okay? And it goes into our cells, into our mitochondria, and O2. Waits for the electrons that are stripped off of sugar, right? Because in photosynthesis, we took the electrons from water and we put it in sugar. Now we're going to take those electrons out of sugar, strip them out, load them up on these electron carriers that then transport the electrons down this transport chain, a second transport chain in the mitochondria. When it goes down, it's got to end up somewhere. That's this yellow line. It ends up back on oxygen. Oxygen takes those electrons back along with protons, right? It takes its electrons back, and then it's going to attract a proton, which is a hydrogen ion. It's taking its electrons back, and the oxygen gas is converting back into what, class? What is this? Water. So in cellular respiration, oxygen gas turns back into water, whereas in photosynthesis, water is split into oxygen gas. This is why one of the products of cellular respiration is 6 molecules of water and 6 molecules of carbon dioxide gas, which happens in the next step, which is the Krebs cycle. So this is a summary, right? We spend $2 to make $4. We spend 2 ATP to make 4 ATP, net gain. And we also load up a couple of these electron carriers. This occurs in the cytoplasm, right? And this is anaerobic. Those concepts will be tested on. Jordan.
On your test online?
Yeah, there was something wrong with the test online, guys. Let me tell you right now. Another teacher created that test. I should have been a little bit more on top of looking at the answers. There was a mistake in 2 of the questions, or at least one of the questions. I have already will adjust your grade for that. Don't worry about that. I must have been bombarded with 1,000 people with the same question, the same concern. I got it. One of the questions that had to do with it's aerobic, so it requires oxygen, and you put that and you got the wrong answer. I got it. We're all over it. And another one has to do with
The other way around.
yeah, carbon dioxide and alcohol, and then lactic acid. Yeah. The word alcohol, the actual alcohol is ethanol.
No, I wrote that.
Ethanol is the name of the actual alcohol itself. There are many alcohols.
Wait, so it's ethanol and what?
Ethanol and carbon dioxide. Alcohol, carbon dioxide, ethanol is the name of that particular alcohol. There are many alcohols. Ethanol, which is the alcohol that's in,
Bars?
hard liquor and beer and wine, that's called ethanol. But we'll get to that in a second. The next phase, right, everything after glycolysis happens in the mitochondria and requires oxygen. So now we're going through aerobic respiration. You see, we've only made $2 from our $40 bill. My $40 bill is glucose, food, and since the cell is like a dollar store and everything costs $1, which is ATP, I need to take my $40 bill, food, glucose, and I need to convert it into ATP. Now, glycolysis, I only get $2 from my $40 bill, so that's not very efficient. But if I have oxygen present, I can go into this mitochondria and get more ATP out of it. Chris?
Well, apparently, one of my partners is saying we really did this, and yeah,
Did what?
these notes, like you've already.
Okay.
Keep going.
All right. Well, like I said before, we're going to go over them again because the test is next class. So it doesn't hurt to go over it.
A big test or a small test?
I don't understand your question. What do you mean, a big test or a small test?
A unit test or like
Just like every other test that you've taken in this class with a Scantron, just the same thing.
Like it's after the packet, right?
Everything that's in chapter 4, no packet. Everything, the whole thing, start to finish. Right?
Okay, thank you.
It's all outlined on School Deed.
Thank you.
And it's also up there. And you'll also get a text. The only thing that you're not getting is me showing up to your house, knocking on the door and saying, hey, by the way, there's a test tomorrow. Okay, Krebs cycle. This is what happens next. Pyruvate goes into the mitochondria if there is oxygen gas. Each one of these expo markers represents a carbon atom that is in sugar. C6, right? Now there is 12 hydrogens and 6 oxygens, but they're not shown here. In glycolysis, outside of the mitochondria, we split sugar into 2 molecules of pyruvate. Each one, if oxygen is present, will go into the mitochondria. As it enters into the mitochondria, we lose 1 carbon dioxide molecule. This carbon is released as carbon dioxide. This 2 carbon molecule, right, bonds with an enzyme, coenzyme A, and forms acetyl coenzyme A, a 2 carbon molecule attached to an enzyme. That's right here. This is the molecule that actually enters into the Krebs cycle. Okay? Then we rip off more electrons, loading up NADH and FADH2. And then when we do that, we release this as 2 more molecules of carbon dioxide gas. So 1 pyruvate, 3 molecules of carbon dioxide gas. We also make 1 ATP in this process. But it happens twice because remember, sugar splits into 2 molecules of pyruvate. So release 1 carbon dioxide, 2 more carbon dioxide. So total, we breathe out 6 molecules of carbon dioxide gas for every 1 molecule of sugar or glucose. And that is represented in the overall cellular respiration equation. But the purpose of the Krebs cycle, for the most part, is not to give off carbon dioxide, that's a waste product. It's to load up these energy electron carrier molecules. Right? That's where I drew up on the whiteboard those empty trucks are when they don't have electrons attached to them, or those extra electrons attached to them. And when they become reduced, or they gain electrons, they turn into NADH and FADH2. High energy molecules. And now they're going to dump those electrons onto the next step, the final step, the electron transport chain. It would make sense that electrons are transported to the electron transport chain. So a real quick 2-minute video summary of the Krebs cycle, named after Hans Krebs, but the actual cycle itself is more appropriately called the citric acid cycle, because right when acetyl coenzyme A attaches to a 4-carbon molecule here, it turns into citric acid. You don't need to know the name of the 4-carbon molecule, oxaloacetate, or citric acid. You don't need to know any of that. All you need to know is what comes in and what comes out. It's very simple.
The oxidation of glucose continues in the citric acid cycle.
This happens if
Pyruvate
oxygen is
molecules
present. This is
formed during
aerobic.
glycolysis diffuse from the cytosol into the mitochondrion. but pyruvate itself does not enter the citric acid cycle. A reaction occurs that removes a carbon atom, releasing it in carbon dioxide. Electrons are transferred to an NADH molecule, storing energy. Coenzyme A, or CoA, joins with the 2-carbon fragment, forming acetyl-CoA. One molecule of acetyl-CoA enters the citric acid cycle. The 2-carbon fragment of acetyl-CoA attaches to the 4-carbon molecule, oxaloacetate, in the first reaction of the cycle. This forms citrate. In a series of steps, bonds break and reform. 2 carbon atoms are released one at a time in molecules of carbon dioxide. Electrons are carried off by molecules of NADH and FADH2. One step produces an ATP molecule by substrate-level phosphorylation. A 4-carbon oxaloacetate molecule is regenerated. Since 2 acetyl-CoA molecules are produced for each glucose molecule broken down, a second acetyl-CoA enters the citric acid cycle. The same series of reactions occurs, releasing carbon dioxide and producing more NADH, FADH2, and ATP. The cell has gained 2 ATPs that can be used directly. However, most of the energy originally contained in the bonds of glucose is now carried by the NADH and FADH2 molecules.
Okay, so at the end of the day, all you really need to know, that's it, all you really need to know is in this cycle, we take in that pyruvate, it goes into the mitochondria, rip off some electrons, make NADH, FADH2, and we release carbon dioxide gas. That's all you really need to know about the Krebs cycle. Now, all these electron carriers, some were produced in glycolysis, some were produced when pyruvate was converted into acetyl coenzyme A, but most of them were produced in the Krebs cycle. All these electron carrier molecules are carrying energy-rich electrons in the form of NADH, that's a molecule that carries those electrons, and I'll actually eat that too, FADH2. They are going to go to the electron transport chain. Now, test question. This goes in the margin. This is what you write in the margin. Where is the electron transport chain located in cellular respiration? Where is the electron transport chain, which is just a series of proteins wedged in a membrane? Where? Who wants to raise their hand and answer that question? Where is the electron transport? Where are these molecules in cellular respiration? Specifically, based on the answer is behind me. So,
Inner
yeah.
membrane.
On the inner membrane
Inner membrane of the what?
of the mitochondria.
You got it. You nailed it right there. This is the mitochondria, 2 membranes. An outer membrane, inner membrane. Right? Outer membrane, inner membrane. The space in between the 2 membranes is the intermembrane space because it's in between them. Just look at them. Look, look. Look at these. This is it. Right here, inner membrane. It is here where the energy from the electrons are going to move down the transport chain only if what is present? Oxygen gas. This guy is going to be waiting for his electrons back. Right? We breathe in oxygen and it goes in through our blood, through the interstitial fluid, diffuses into our cells and into the mitochondria, just sitting here. You know, millions and millions and millions of oxygen atoms sitting here chilling. And the electrons will move down and bond to oxygen gas, turning it back into water. Okay? So this will only happen if oxygen is present. And when the electrons move down there, it activates a pump. That pumps protons from the matrix and crams them into this space right here. We are cramming protons in here, creating this dam, this buildup, this proton gradient of high concentration that's going to fuel this enzyme called ATP synthase. As the protons pass out, as you'll see in the next diagram, actually not this diagram, the next slide.
Good ass diagram.
Okay, so this is a mitochondria. Look, here is the electron transport chain in the inner membrane space. We're going to pump protons from here, the matrix, into, right, this space right here, the inner membrane space. You all right there? Giggles, McGiggleson?
Yeah, I'm fine.
You're right.
What about this room?
I'm fantastic.
You seem a little pissed off right now.
No, I'm not pissed off. By the way, videotaping this. Watch your mouth. Thanks. I hope all of that gets recorded.
Mr. Beard, can you hear our conversation?
Yes, I'm pretty sure you can hear your conversation. Can't wait to hear it back.
Alright, Ashley, you can come back.
Alright, this diagram is on repeat. This is a gift. You guys done? Are you done?
Oh,
I'll wait. That's the next step. This is the beginning of the electron transport chain. The molecules that transport the electrons, NADH, FADH2, they're electron carriers. NADH, when it loses its electrons, it goes back and turns back into, look, NAD+. The energy is in the electrons. And it's pumping hydrogen ions, right? Here's the energy right here. The electrons. Hydrogen ions are just protons. The energy is moving down. Look, it's pumping protons. It's losing energy every time it does that. Going, pumping protons. It's got to have a final place. The final electron acceptor, like I said, oxygen gas, combines with oxygen gas and makes water. So now that we have all these built up protons, they're going to pass through ATP synthase, and when they do, this enzyme attaches a phosphate to ADP, making ATP. This is why we eat and breathe. Pretty important. We eat to get the energy electrons from sugar to pump this stuff in here. We breathe so that there's oxygen to accept the electrons back. So when we pump these things in here, they will pass out and make a bunch of ATP. How many ATP does this process make? About 34 on a perfect day from every molecule of sugar. So my $40 bill makes $2 in glycolysis, $2 in the Krebs cycle, but the other $34 happens here. This is where the real magic happens. So I go to my bank, and I'm like, here's my $40 bill. Here's my bank would be like mitochondria. Here's my sugar molecule. I need to convert this into ATP because the cell store only takes dollar bills, only takes ATP. He gives me 38 $1 bills back. I'm like, it's not bad. I'll pay my $2 heat transaction fee if oxygen is present. So, here's your summary in terms of energy. If oxygen is present, we make 2 in glycolysis, 2 in the Krebs cycle, 34 here. 34 on a good day. No, every single molecule of glucose can make 34 ATP. There are billions of molecules of glucose in your body. Billions and billions, trillions, right? An astronomical amount. Per second. You know what I'm saying? You have like 4 or 5 seconds worth of ATP in your body. Let's say you had 0 glucose in your body. Your ATP would last you about 5 seconds. Then you would be dead, done. You would be completely dead. Except we don't. Guys, let me see the cell phone. Sam, where am I this? But you have glucose stored in your body so that we can recharge that ATP, right? As it turns into ADP. We can turn it back into ATP. So this happens as long as we're still delivering oxygen gas to our body. This is where the respiratory system and the cardiovascular system come in. We're constantly supplying our cells with oxygen gas so that we're always doing this. So the final video is the final step of cellular respiration. This is 2 minutes long, 2.5 minutes. And this gives you kind of a summary of everything.
Most of the energy harvested from organic molecules during glycolysis and the citric acid cycle is stored in NADH and FADH2. These molecules give up their high-energy electrons in the third phase of cellular respiration, oxidative phosphorylation, where most of the cell's ATP fuel is produced.
The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondria. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward highly electronegative oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in
Those are the
electron
electrons.
transport.
That's
The
oxygen
last
gas.
complex adds a pair of electrons to an oxygen atom and 2 hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse through a special protein in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. This mode of ATP production is called oxidative phosphorylation because it is powered by the transfer of electrons to oxygen. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by electron transport and oxidative phosphorylation, about 34 ATPs for every glucose consumed.
This illustration summarizes cellular respiration. Glycolysis and the citric acid cycle produce a small amount of ATP via substrate-level phosphorylation, but most of the cell's ATP is made via oxidative phosphorylation, when NADH and FADH2 produced in glycolysis and the citric acid cycle give up to oxygen the electrons obtained from organic molecules.
Okay. At any point in time, if you ever get confused and you want to go over, there's nothing stopping you from looking at the playlists I put on Schoology and just watching these videos again. They're only a couple minutes long, right? There's like 36 of you in this class. More. It's probably almost, there's like 40 of you actually.
37 and a half. Wow.
Only 13 of you did the homework assignment last Friday. Homework and read. The reading.
Wait, can you do it like
You were
Saturday
one of the
night,
30.
not like Friday night?
Now listen.
I said it's in Jordan.
Oh yeah, I most definitely did it on at 11 o'clock.
There are things that you need to do on your end as a student to prepare for the exam. A, pay attention to class when I lecture. But that's a small portion, right? B, are you actually doing the reading? Are you actually opening up these files right here? Right, when you go to a homework assignment, for instance, I'll just go to, I don't know, homework number 4. So, are you opening up these files and reading? Now, you don't read this like it's, you know, the novel Twilight. You don't just start reading from the beginning to the end. You will bore yourself to tears, and you will not retain enough information for it to be useful. A science textbook, you start off number 1, you flip through the whole thing, look at nothing but the pictures. You only look at the diagrams. So I'm going to look at that, look at those pictures, and then I read just this little part that is associated with the pictures. Go to the next, I look at that diagram, read that right there. Look at this picture, read that. Look at this picture. Look at this picture, read that. Look at that picture, read this. Look at this picture, read that. Done, done. That took me a minute and a half, right? If I actually look at every minute and a half, done. Second step, go back and look at just the bold words and the sentences in the bold words. And when I say bold words, I even mean the headers. Cell diversity. Okay, there's really no bold words there. Cell shape. Okay, really no bold words there. I'm going to skip to the next thing that has a bold word. Cell size, nothing. Here we go. Basic parts of the cell. Plasma membrane. Read the sentence that has that in it. Cytoplasm. Read that sentence and read that sentence. Nucleus. Read that sentence. Prokaryotes. Read that sentence. Eukaryotes. Read that sentence and that sentence. I'm going to read all those sentences right there. Okay. Maybe any italicized words might be important. When I'm done with that, I'm going to try and answer the questions. Try and answer the questions. If you can't answer the questions, you need to go back to the header that had that information and read a little bit more. You shouldn't take more than 10, 15 minutes on doing this. You do not, I repeat, do not, in a science textbook, go to page 1 of the reading and just start reading. Your brain is not programmed to process information that way. Especially when it has to do with all these words that you may never have seen before. Especially when you're doing like the last homework assignment that you guys did, which was homework number 8. Right, homework number 8. Scroll down to the very bottom. Did you guys know that if you do this, watch, all materials, tests, bam, all the quizzes go like right there?
Oh, I didn't know that.
I want you to respond like, whoa. Oh, I didn't know that. Now you do. Also, if you go and click and just click on the assignments.
Can't do that on
iPads suck, guys. Don't use them. I would only access Schoology on my computer. The fact that you guys have iPads, but here you go. This is how you do it. You don't need to. It's all in order anyway from the time that I put it. But look, if I click on this, these are the people that did it. Yeah, is that 36 people? I don't think so. See how I can tell if you're late right there?
Yeah.
Don't worry about it. I'm not going to mark you guys late because I haven't even inputted. I just inputted it today, so it's fine.
Are you gonna put?
You only have to do these 4 questions into this section. You don't have to do the critical thinking. But look, once again, guys, the first thing I'm doing is I'm looking at this picture. I'm looking at this picture. I'm reading this picture. Right? This picture right here. That right there, I'll probably look at. This right here, read that right there. This guy's making some cheese. What's that all about? Read that. Alcoholic fermentation. Yummy, yummy. Read that. Yeast. Read that. Oh, that looks important. Done. Then I go back and I read all the sentences that have what in them? Bold. Anything big and bold, right? The titles of the sections and the bold letter words within them. Overview of cellular respiration. Bam, I'm reading all that. I'm reading those sentences. That's how you read a science textbook. Not just in high school, not just in middle school, all the way up through college. That's what I did. Okay? That is a reading strategy. You do not treat this sort of informational text in the same way that you would treat a novel, an article, a newspaper. Okay? It's totally different. Got it?
Got it.
Good.
Are you gonna import this grades in?
Quiz grades for anaerobic and anaerobic, they probably won't be in this one. They'll probably be in the next grade. But your photosynthesis quiz will be on there. You guys were here, right? Questions. Question. Know this equation. Memorize this. This is the what equation? Equation for what? Yeah, you need to know that these things right over here, these things right here are called the? Also, a little bit of energy sometimes is released. Energy is released and placed into ATP, right? So, energy is not really a reactant in the sense that it's not matter. Right? It's kind of like saying, I'm making a 6-egg omelet, cheese omelet. I need 6 eggs and some cheese, a cup of cheese. Those are my reactants. And then chemical reaction happens, I cook it. And my product is 1 6-egg cheese omelet. Now, is heat really an ingredient?
No.
No, really, it's not in my omelet. I'm not eating bites of heat. Heat was part of the process that was added into it, right? So energy is not really technically a product, but you can sometimes, depending on the textbook, see that energy is either released. If it's released, it's in the right side of the arrow. It's what is produced, right? We're releasing energy. If it's absorbed or placed into the chemical reaction, as is with photosynthesis, it's going to be a reactant. It's going to be on this side. So photosynthesis, we put energy in to sugar. Cellular respiration, look, sugar's over here. We release energy out of sugar. Energy is released. That's what's produced. And it's used to make ATP. So there will be test questions comparing the two things, right? Photosynthesis, energy storage, cellular respiration, energy release. This one happens in a chloroplast, this one happens in mitochondria. And then their chemical reactions are reversed. So the reactants of one, look, products of the other. The products of one, reactants of the other. Pretty simple, right? Understand that relationship. So we'll take a 2-second break from this. We'll We'll come back to fermentation. You guys remember this diagram right here?
Yeah.
This is what you were supposed to do when I was absent.
What? No, you didn't hear here.
Right? You were supposed to do this when I was absent. The function of photosynthesis. So take out that worksheet. Double check. See if you guys got the right stuff. Photosynthesis. This is just a comparison of the overall processes. Stores energy, releases energy. Location, chloroplast, mitochondria, and then the equation. Notice, guys, I don't even need to write over this. If I know that it's just the reverse, right? I don't really need to, as long as you understand that it's the reverse, right? This is 6 molecules of carbon dioxide, 6 molecules of water. Obviously, plants take in carbon dioxide and water. That's something we learned in 5th grade. And they produce sugar and oxygen gas. The reactants are the things on the left of the arrow. That's what we start with. The products are what are produced. We produce something at the end. That's right here. And cellular respiration is reverse. We don't consume carbon dioxide and water in cellular respiration. We produce it. We don't produce glucose and oxygen gas. We consume it. Okay. Here's where students get a little confused. There will be a test question about autotrophs and heterotrophs. So know the difference between those words. Auto means troph refers to
Food.
feeding. So if I'm a self-feeder, I'm a plant. I'm a producer. I make my own food in my chloroplast. I don't need to eat. But this is where students get confused. It's where we talk about the mitochondria. People are like, oh yeah, animals have mitochondria, plants have chloroplast. No. Plants have mitochondria too. You see, just because they made their own food doesn't mean that they don't have to also break it down and put it into ATP, the energy into ATP. So plants, the minute they make sugar in their cells, it goes right into a mitochondria and gets broken down to make ATP. So both autotrophs, plants, and photosynthetic bacteria and algae and other protists also have mitochondria. Heterotrophs, right, us, we cannot make our own food. We only do this. So the way that we get our glucose to start with is by eating, by consuming. That's why we're also called consumers. So to start off with photosynthesis, this right here, guys, is all from this diagram in your notes. Right here. Now look at the arrows. Look at the arrows. What are the reactants of the light dependent? Well, first of all, this is not a reactant because light is energy. That's why it's a wiggly line. Look at the 3 arrows that go in. Right? If you go into something, that's what you start with. These are the reactants. But water, ADP plus, low energy. ADP, low energy. You're going to get charged up by the light. Energy gets stored in. Energy storage. I'm in a chloroplast. This is in the thylakoid. Electrons are going to fly off, right? Water gets split. We still off the electrons. Okay, we're going to pump hydrogen ions from the stroma into the thylakoid space. They pass back out to ATP synthase to make my products. When I split water, I produce, look at the arrows coming out, oxygen gas. ADP gets charged up to ATP. NADP plus gets charged up to NADPH. These are the products. Now that is just talking about light dependent. This only occurs during the sunlight. Light independent occurs during the sun and without the sun. It doesn't have anything to do with sunlight. This is the Calvin cycle, also known as the light dependent reactions. What goes in? What are my reactants? Carbon dioxide gas comes in through the leaves, through the stomata. And we're going to unleash all of this energy onto carbon dioxide to make sugar. Another word for sugar would be what? Sorry. There's the same picture, right? Also, O6. That's C6H12O6. Right? So ultimately, when I look at this, that's what that is. That's a summary of all that. Light dependent, you need the sunlight. It happens in the thylakoid membranes. That's what that picture showed. And these are all the 3 things that were the arrows going in. These are the 3 things that the arrows coming out. The reactants are the things on the left, the products are the things on the right. The products, right, ATP and NADPH, the energy products, are actually what fuel the dark reactions. Not dark reactions, light independent reactions. This happens in the stroma. This is the Calvin cycle, also known as the Calvin cycle. Carbon dioxide goes in, we take the energy from ATP and NADPH, put it into carbon dioxide, and we make sugar. ATP goes back to ADP, NADPH goes back to NADP+. This stuff, guys, just leave, you don't need to write in the reactants, as long as you understand which one's the reactants and which one's the products. You know what I'm saying? Save yourself some time. Can I move forward?
No,
I'll give you guys about 20 seconds.
we're about 30.
Okay, ready? Cellular respiration. We just went over all of this. Glycolysis occurs in the cytoplasm, makes 2 net ATP. 2 net. Right, it's a gain. It actually produces 4, but it's net. We start with glucose. And it splits into 2 molecules of pyruvate, otherwise known as pyruvic acid. Does this require oxygen? No. This is also going to go into fermentation, which we'll learn about in a second. Now, if we do have oxygen, right, then we can go into the mitochondria, make a whole lot more ATP. That molecules of pyruvate actually, pyruvate technically doesn't go into the Krebs cycle. It gets converted immediately into acetyl-coenzyme A. We release a little bit of carbon dioxide, and eventually through the Krebs cycle and the electron transport chain, we make carbon dioxide and water and a bunch of ATP. Got it? Now what happens if we don't have enough oxygen gas? I'm going to give you a scenario, and this is relative to everybody here because everybody's experienced this at one point in their life. They probably experienced this today if they had PE already. Did they have PE already? You stare at something and then you have this like spot in the corner of your eye for a while. I don't know what it is, but it's like. Okay, let's say I'm running uphill. You see, if you're jogging normally and you're being able to provide enough oxygen to your muscle cells, like you can breathe nice and normal, you can run for a long time. Because your body is going through aerobic respiration. You're getting enough oxygen to your muscle cells. You are burning 1 molecule of glucose, $40 bill, and you're making 38 ATP. This is good. This is efficient. This is aerobic respiration. Your lungs are delivering oxygen to your muscle cells at the rate in which the muscle cells are demanding oxygen. But let's go uphill. Let's run up a hill, right, with a backpack full of weight. Now, your muscle cells are demanding a lot of ATP per second. Whereas before your lungs could provide enough oxygen, now it cannot. The scenario will go like this. Let's say that if you're running uphill, your muscle cells are demanding, let's say 1 muscle cell is demanding, I'm just going to give an arbitrary number, 1 billion ATP per second. Your muscle cells need 1 billion ATP per second, but your lungs and your heart and your blood can only provide that muscle cell enough oxygen to make 500 million ATP per second. Your muscle cells have a problem. They need 1 billion every second for this guy to run up a hill. But because of the condition that you're in, your conditioning, your lungs, your blood, your oxygen supply, right, can only make, through this process, 500 million. You are short 500 million ATP. Well, your muscle cells are going to make that other 500 million ATP, no matter what, because your brain is telling your muscles to fire, and you got plenty of sugar stored in your cells. So we're going to burn that sugar still, but instead of getting $38 with oxygen for every one $40 bill, you're going to go through that process, glycolysis, which only makes how many ATP for every molecule of sugar? 2. You're going to burn it without oxygen because this process doesn't require oxygen, but the problem is that when you split sugar here, NAD plus turns into NADH, all of my electron carriers get full of electrons, and they have no place to dump their electrons. You see, because before, these guys dumped their electrons over here and ended up on oxygen, but like I said before, we don't have enough oxygen. So this whole process will stop. Your body will not be able to produce any more ATP. You will not be able to climb up that mountain unless we switch over into anaerobic respiration. Guys, eyes up here. Eyes up. Anaerobic respiration is a way for cells to continue this process without oxygen. This guy needs to unload his electrons, so he turns back into this and allows glycolysis to continue. So what he's going to do is we're going to dump those electrons back onto pyruvates, and in muscle cells, we make lactic acid. So lactic acid starts to build up. But you know what? At least we're making 2 ATP each time. 2 ATP, 2 ATP. Lactic acid starts to build up in our muscle, and it causes that burning sensation. And eventually, if too much lactate gets built up in our muscle, it causes muscle fatigue. And the only way to clear that lactate is to introduce what into the cell? Oxygen gas, which will take those electrons back off of lactate, turning it back into pyruvate. And then you can just have that pyruvate go into the Krebs cycle. So that's the final 3 or 4 slides. He's talking about what do we do when we don't have enough oxygen. Here.
You look stressed. You need to.
I don't need a diagnosis of my mood from you or what's wrong with my eye.
I'm just stupidly stressed. You have to get all like pissed off about that.
Don't.
I'm trying to look after you.
We're gonna have a little chat after class
Oh, see.
about you telling me that I'm pissed off. Okay?
Do I just stand there?
Yeah, hang out. Yeah, hang out after class.
I
We're
gotta
gonna have a little chat.
Hey man, what's happening? You can't eat it. I'm here. I get it. Bring it out.
Apparently, I was very mad.
Just watch my rap.
Okay. Pass out. All right. Fermentation is anaerobic. This is a process that makes 0 energy, but it allows glycolysis to continue when there's no oxygen. This concept is so hard for students to grasp. Fermentation doesn't make energy. It takes electrons from NADH and makes NAD plus again. Okay. Okay. Read this. The key's right there. Read that. You only have so many NAD pluses in a cell. When they're all loaded up to NADH, here's what happens. Okay? In a muscle cell, here's what's going to happen. We're going to split sugar through glycolysis to make 2 ATP. Now, this doesn't require oxygen, so we can make energy from glucose without oxygen. But all my NAD plus gets loaded up in NADH. Then we run out of NAD plus. It's all loaded up. All of our trucks are loaded up with electrons, no place to go, because there's no oxygen. So we have to unload. We have to unload this truck so that it can be unloaded and go back so we can split more sugar and make more ATP. The process that unloads this is called lactic acid fermentation. If you're in a muscle cell, we unload it right back onto the pyruvate. It's a 3-carbon molecule. When it accepts those electrons, when it becomes reduced, it gets converted into lactic acid. Why is this a problem? Lactic acid builds up in our muscles, causes muscle soreness, causes muscle fatigue. Got it? So, ultimately, lactic acid is where we take the energy from NADH and we convert pyruvate into lactic acid, and NADH is converted back into NAD+, allowing us to make ATP. Is this aerobic or anaerobic? Class? Once more time. Is this aerobic or anaerobic? Class? Anaerobic. There we go. I'm going to show you an episode of a TV show called Stanley's Superhumans.
Stanley's awesome.
Congratulations.
Was this about the lactic acid guy?
Yes, you've already seen this, so you get to watch it again.
Damn.
This guy is a freak of nature.
Yes.
I have seen this clip probably 35 times.
I've seen it twice.
As humans, we've been running for over 4 million years. The ability evolved to help us hunt prey, transforming the anatomy of our ancestors. But since then, biologically little has changed. Even the marathon runners of ancient Greece, who were famous for their long-distance runs, could not run forever. However, I've arranged to meet an endurance runner whose remarkable body may have taken the next step in human evolution. That's because Dean Karnassus is a man some believe has a genetic mutation that allows him to run forever. Earlier this morning, Dean began a 12-hour dawn till dusk ultra marathon. To catch up with him, I've taken to the air over beautiful Marin County, where Dean has told me I will find him running along the quiet country roads.
I think I found him. There he is. That must be him.
I've heard Dean has superhuman stamina, and one day started running and hasn't stopped. He's run 146 miles through Death Valley in 130-degree heat, and once even ran an ultra marathon to the South Pole. I want to know how Dean's muscles can keep going longer than anyone else on Earth. In my search for superhumans, I've asked to witness him undertake this mammoth 12-hour run. So far, he's been running for 2 hours, ran nearly 16 miles, and will attempt to keep going until the sun goes down. And against my better judgment, I'm gonna run with him for as long as my body can handle.
You just put one foot in front of the other until you either cross the finish line or you pass out in the process.
I want to find out how Dean is able to conquer such a physical and mental challenge without suffering fatigue. But before we set off, I've called in Joseph Smith, a sports scientist from the University of California, San Francisco, to observe.
Got any advice for Daniel?
Pushing your body to that extreme can be, can be dangerous and even fatal.
To make sure I don't harm my body, Joseph will follow us in a support vehicle. But I've also asked him to monitor and observe Dean's progress. And as we settle into the run, Dean explains how he became one of the world's greatest endurance runners.
Well, I used to love to run when I was a kid, and I ran all my life up until I was 14, and then literally stopped running. Went to college, then I went to business school. I had this really cush corporate job, everything that was supposed to make me happy, and I was freaking miserable. I hated my job, I hated my life. So, on my 30th birthday, I'm in a bar with my bros, drinking bad tequila, And I said, hey guys, I'm leaving. I'm like, I'm gonna go run 30 miles tonight to celebrate my 30th birthday. So I walked out of the bar. I didn't even have running shoes, I had gardening shoes on. And I thought, this is nuts. I'm just too drunk, what am I doing? But it felt so right. I couldn't believe how good I felt. And that night changed my life forever.
It's amazing to think a single moment transformed Dean from an average guy to a potential superhuman. And only 40 minutes into the run, It feels like my body is about to collapse.
I'm really trying my hardest, Dean. You're doing great, man. No worries. You've been going a long time. You're doing great. Dean, the whole point of my adventure here is to find superhumans. And I really feel that I'm holding you back right now. But I really want to see what you do. I really want to see what you do. I'm gonna have to wuss out, man. I'm gonna have to get in the car with Joe. You're not wussing out at all. That was fantastic. Thank you. No, that was fantastic.
Such a mean person.
Did you see the way he ran?
I just couldn't do it. My legs started cramping up, my lungs were burning, I mean, my heart rate got
Another guy
over
was waiting,
200
right?
beats per minute constantly. I'm exhausted, and as I watch Dean settle into his stride from the comfort of the car, I can't believe he's going to keep running for another 11 hours. No matter how fit you are, demanding exercise will eventually exhaust the human body and force you to stop. This is mainly due to the fitness levels of our heart and lungs, but there are other biological factors that should eventually stop us all. Levels of lactic acid, a byproduct of our muscles burning energy, quickly build up in our blood during extended physical exercise. This causes the muscles to become fatigued, cramp up, and force you to quit. But as Dean continues to run, amazingly nothing seems to be affecting his pace.
I've never witnessed anything like this before. What we're seeing here today, it's just extraordinary, and it's actually very beautiful to see him run. You know, we've been following him all day here, and his mechanics still seem very solid. He doesn't seem to be getting fatigued at all, in fact, and he seems very comfortable with his pace. It seems like he could just go on forever.
In 2005, Dean shocked the science world by running continuously for an astonishing 80 hours and 44 minutes, the equivalent of over 10 back-to-back marathons. And in 2006, not only completed 50 marathons in 50 days, while other athletes
He
would suffer
ran
muscle
4 days
fatigue,
straight without
amazingly,
sleeping.
Dean's pace got faster, and he even decided to run the 1,300 miles back to the start. But how is this possible? The reasons why Dean is able to run such incredible distances without stopping is a complete mystery. But one thing's for sure, is Joe and I watch him ease to the final stages of his 12-hour run. His mental determination is truly inspirational. Finally, Dean's breathtaking dawn till dusk run comes to an end.
I've never witnessed anything like that in my life. You ran for over 12 hours. I couldn't even keep up with you for just a couple of hours. I mean, well, that's what I love to do. My legs were burning, my chest was aching, but then that's what I do, I run.
Dean has effortlessly completed the 12-hour challenge, but even after his ultra-marathon, instead of riding in the car with us, he decides to run all the way back to San Francisco. And tomorrow, Joe and I plan to find out if Dean's body can really run forever.
He's been running now for 24 minutes. The last 2 readings are actually going down.
And what we discover is truly astonishing. My name is Daniel Browning Smith, and I'm a superhuman. Comic book legend Stan Lee has sent me on a mission to find other superhumans. My search has brought me to California to meet ultra-marathon man Dean Karnazes, an endurance athlete who appears to be able to run forever. Sports scientist Joseph Smith and I have already witnessed him complete a grueling 12-hour run.
I've never witnessed anything like this before. It seems like he could just go on forever.
But whether or not he actually has a superhuman ability is still a mystery. Now at the University of California, San Francisco, Joseph and Dr. Zintas Ahrens are putting Dean through a series of high-performance fitness tests.
So we're going to be working Dean about at an 8 miles per hour pace.
The doctors want to find out if Dean really can run forever.
This guy's a raw specimen. Already at 1.15. This is amazing. Already 163.
Hold on a couple minutes and then they stop.
And if so, the secret behind his incredible endurance.
The side clip, we can really see a transition of his heel strike and running mechanics, so we can see he's very solid and very strong there, which probably allows Dean to run for very long distances.
So far, the tests show Dean's fitness levels match those of Olympic runners. and the biomechanics of his body confirm he was built to run. However, there's nothing to suggest Dean is superhuman.
Okay, so let me get I'm going to take a baseline of your blood lactate level as well, okay, Dean? Sure.
In search of answers, the doctors decide to perform a lactate analysis test.
How you feeling right now, Dean?
6.
The test will measure the buildup of lactic acid in Dean's blood as Joseph and Dr. Zarings deliberately increase the levels of physical exertion.
Are you going to jump off there, Dean?
Lactic acid is a byproduct of our muscles burning fuel and energy from the body.
How are you feeling now, Dean?
I would say about 8.
As the levels of lactic acid increase, the more fatigued our muscles become.
How are you feeling now, Dean?
I would say I've gone up to about 10 this time.
Okay.
They tire, begin to cramp, and ultimately render the human body incapable of taking another step.
How are you feeling now?
Maybe 12, maybe 13, maybe 12. 12.
12?
Yeah.
But as the test continues, the doctors notice something unexpected.
Lactate levels are not rising very much, which is very unusual, even though we're increasing the intensity.
And things soon become even more extraordinary.
This is absolutely amazing. His lactate levels are not rising. Usually you would see an inflection and a rise as we increase exercise intensity, but he's actually clearing that lactate.
Amazingly, despite the increase in exercise, it seems the levels of lactic acid in Dean's blood are not rising.
He's been running now for 24 minutes, and we've been getting a blood lactate data point every 3 minutes. And the last 2 readings are actually going down.
This is extremely unusual and goes against everything we understand about human biology.
2.1.
2.1.
2.1.
So did it just drop?
Just drop again.
Strong.
So how are we going to get this guy's threshold? He's clearing that lactate. We're trying to wait for an inflection.
At this pace and intensity, most people are up higher than 4.
Yeah. Can't watch the video.
This is a totally superhuman
Yeah.
experience we're having right
Yeah.
here.
Really, he can run forever because there's nothing stopping him. His lactate levels are now
How you feeling now?
lower than they were in the very, very beginning of the test. So it's amazing.
Incredibly, it seems we've uncovered the unique physical power that allows Dean to just keep on going. His extraordinary ability to reduce the level of lactic acid in his blood defies human biology and shows he could really run forever.
Have you ever seen results like this before?
Never.
Never? Have you?
No.
I don't know if that's making me feel good or bad.
It's amazing.
Okay.
He's just running in the corner.
That is lactic acid fermentation. But there's another type. Not done by muscle cells, but done by yeast and other types of bacteria. I'm going to tell you 2 little stories. First of all, if any of you guys get arrested and go to jail, some of you will, okay? You know who you are. You're going to spend some time in jail, right? They don't serve alcohol in jail, so obviously you're a criminal. Somebody there knows how to make alcohol. They've learned because they've been there for like 20 years or whatever it is. So you're going to want to make buddies with that guy. Hope that you share a cell with him or her, okay? So what they make, to make alcohol in jail, they call it hooch, right? Well, I'm going to tell you what hooch is. First of all, you need to make some alcohol. You need a source of glucose, right? You need some sort of juice that has sugar in it. You know, we make tequila from the juice of a blue agave cactus plant. We make gin from the juniper berry. We make whiskey from either wheat or corn. We make wine from grapes. We make beer from barley and hops. It all starts with some sort of plant that goes through photosynthesis because it starts with glucose. And then we add an organism that goes through anaerobic respiration in order to make its ATP. You see, these organisms don't make lactic acid. They make alcohol and carbon dioxide gas. So we add a little yeast. But since I'm in a jail cell, I don't have access to yeast. The first thing I need is my juice, so I'm going to go sneak in some grape juice from the commissary or whatever, from the cafeteria. So somehow you had to sneak in some grape juice into your cell. And I need a bowl, right? So I'm going to, what's the only bowl that's in your jail cell? Toilet bowl. So I'm going to dump in all that grape juice into my toilet bowl, right? So I have my source of glucose. Now I need some yeast. Where can I find some funky yeast? Your feet. Okay, so I'm going to take some stink-ass socks that have been just funky for like 6 days. I'm going to toss those bad boys right there into the toilet bowl. I'm going to let those puppies just sit there and ferment for a couple of days. Right? As it's fermenting, the yeast cells are taking in the glucose from the grape juice, making ATP for itself. But in order to continually break down sugar, it needs to unload NADH into NAD+. And what it does is it dumps those electrons, making the pyruvate and literally producing alcohol, specifically ethanol, and carbon dioxide gas. So I look at my toilet bowl and it starts to bubble. What is the bubbling? Look at behind me. What is the bubbling in my toilet bowl after I've added my yeast? Yeah. Carbon dioxide gas. If I blow a gas into water, it forms a bubble. Try it sometime. Okay, when you're doing, Chris. Shh. Scenario number 2. Obviously now I have to drink this stuff.
Yep. Bought it.
I need to know if it works. Go to jail. Figure it out. Let me know how to hooch this. Let's review our explanation. Don't go home and start putting like your brother's grape juice in the toilet bowl and finding your dad's stinky socks and trying to make your own booze and drink that stuff and be like, well, Mr. Bureau said, no, don't. It will not work. It will taste like crap. It takes a very highly trained criminal that has been doing this for years. I have no idea. I've never made hooch. I don't plan on making hooch. But my mom, when she came to this country, they used to make their own bread, right? And to make your own bread, you start off with flour and eggs and stuff like that. The flour has all the glucose in the starch. And so you add yeast to the bread. Does anybody know why we add yeast to the bread? Rise. Is it magic? No. I add this organism, yeast is the single cellular fungus, and then I mix it into the dough, and all of a sudden it starts to inflate. It starts to rise. What is causing the dough to rise? What is it? Carbon dioxide, because the yeast is going through alcoholic fermentation in the bread. I know what your brain's thinking. Hold on, Mr. Greer. Are you telling me that when my mom or dad makes bread, that it's also producing alcohol in the bread dough?
Yes.
Yes, it is. So why am I not getting drunk every time I eat bread?
The oven heat's not going to
I think the word you're looking for is evaporates.
Yes.
Yes, correct. You're going to bake that bread at a very high temperature, which would obviously evaporate the alcohol. You're not going to actually taste any alcohol or get drunk from any alcohol that was created through the process of alcoholic fermentation when you added yeast to the bread. So, same thing. Anaerobic respiration does not require oxygen, totally different organism, totally different products. You're not making lactic acid, you're making alcohol, which is a 2-carbon molecule, and that 3rd carbon that came from pyruvate is released as carbon dioxide gas. So when you make wine, it bubbles. You got to siphon it out. And then you got to kill the yeast at some point. Actually, if the alcohol percentage gets above like 14-15%, it becomes toxic to the yeast itself, or the organism that's causing the fermentation to occur, and will kill it. They're just sitting there swimming in their own alcohol, and it's toxic to the cells. It's cytotoxic. We're done with our notes, right?
Yeah.
Yeah, finally.
Let's do art clickers.
We're going.
Oh, and now?
Extra credit.
Yeah.
Now remember that worksheet that you guys had? Are you missing a chart?
Yeah. The last one. I know that was helpful.
So the chart should look like this. Lactic acid occurs in the muscles. Does fermentation produce any ATP?
Yeah. No.
It allows glycolysis to continue without oxygen, and it's in glycolysis where we produce 2 ATP. Starting molecule for both is glucose. It's the ending molecule that differs. With lactic acid, we produce a 3-carbon lactate, right? Lactic acid is 3 carbons. 1, 2, 3 carbons. Ethanol is 1, 2 carbons, and carbon dioxide is where that 3rd carbon comes from. Is oxygen required? Hell no. This is both. These are both anaerobic. What? Come back in 5 minutes. Gio, did you hear me?
Wait to make two.
So the purpose of glycolysis is not to make energy, it's to unload NADH into NAD+, date time, and allow glycolysis to continue to go. It's now very efficient. I'm only getting $2 for every $40 bill. It's actually the best way to lose weight, right? Because you're burning way more sugar this way to get the same amount of ATP. Because you only get 2 ATP per molecule of sugar. So in order to get like 100 ATP, you'd have to burn 50 molecules of glucose. In order to get make 100 ATP with oxygen, you burn much less. You burn much less, only like 3 or 4. All right. You guys also remember that molecule, or that molecule. This worksheet. Remember this worksheet right here? There. Let's go over this real quick. So we'll start for here. Glycolysis starts with glucose. And it gets converted and split into a 1, 2, 3 carbon molecule. Right? A, it has 6. Gets split into 2, 3 carbon molecules, which is B. What's that?
Pyruvic.
Some of you guys put pyruvic acid. It's the same thing. Now, in this step from pyruvic acid, when it crosses into the mitochondria, it gets attached to this coenzyme A, right? Oops. Coenzyme A. This 3 carbon molecule loses one of its carbons as this gas. This is a little gas molecule, right? And then now it's only a 2 carbon molecule. That's what C is. This is acetyl coenzyme A. It's acetyl coenzyme A that goes into the Krebs cycle. Now the Krebs cycle is where we rip off the electrons to produce this D, FADH2, about 2 molecules of that. But look, E is produced here in glycolysis. It's produced here when we make acetyl coenzyme A from pyruvate, and it's also produced in the Krebs cycle. E goes into the oxidative phosphorylation. What's the other electron carrier? No, it's loaded up. Now G is a gas as well. That's my little gas cloud. What gas goes into the electron transport chain? And it turns into, when it accepts the electrons from E and D, it turns into this molecule right here. Right? Now H is, what are we ripping off of sugar here and ripping off of sugar here? What gas is H? Right? Begins with glucose, broken down through glycolysis, net gain of 2 ATP, occurs in the cytoplasm. We produce 2 molecules of pyruvate. If you have no oxygen, right here, no oxygen, you go through fermentation. There are 2 types of fermentation. Alcoholic fermentation, where we produce alcohol and carbon dioxide, and then we have lactic acid fermentation, where we just produce lactic acid. If you do have oxygen gas, which is this right here, that pyruvate gets broken down into acetyl coenzyme A, and we go into the Krebs cycle. Krebs cycle is located in the inner membrane of the mitochondria. You produce some FADH2s, some NADHs. You also make a little bit of ATP. That's going to be used in the electron transport chain to make a bunch of ATP, 36 to be exact. Did everybody do their warm-up number 11?
Yes.
Number 1 through 11.
Okay, so I told the sub to have them do the warm-up first. I told the sub in an email to have the kids take out a piece of paper and write warm-up number 11 and number 1 through 11. And then fill in 1 through 11. If you have not done that, then you should take out a piece of paper and just write 1 through 11, title it warm-up number 11.
I have a question. I want you to take right now. Yeah, the sub, I swear, was talking about the Clash of Clans the whole period. Yeah.
Absolutely.
He was bragging that his village was better than everybody else.
Okay, so those two worksheets that you guys just went over here go in your binder under work. They will be checked in the next binder check. No, next couple of weeks. Today. I'm going to update some stuff. You'll see your, if you guys did not do a binder check and you never got, like you were absent that day and you never had anybody grade your binder, you're going to get a 0 out of 140 or 140. Okay? And that's going to dramatically affect your grade until you make that up. The way you do that is you have somebody else grade your binder check. Use one of the rubrics that are in that are in that what's it called? And then have somebody grade it, and then come and give me the graded binder check with your binder, and I'll check it. Until that happens, you get a 0.
Now we're, oh my god, you're on the bad website, man.
It's just Dropbox. Every time it syncs with something, it flags as if there's some sort of threat, when all it is a temporary
What about
file.
YouTube? Like, I'm on YouTube and it says like malware blog. Like,
Because there's ads that pop up during that YouTube video.
okay, like I didn't know.
That's what's malware.
Okay, thank you.
All right, number 1 is a process. We start with process outside of the mitochondria. That's glycolysis. That's what I'm doing right now. That's exactly what I'm doing. I'm not going to go real quickly. I'm going to go at my pace, my speed. Hopefully you can catch up. You should have already had this done. Number 2 is a process. That's the Krebs cycle. Everything that's up here is written on the whiteboard over there. There's nothing that's supposed to be filled into these blanks that's not somewhere on that whiteboard, or in your book, or in your notes. The last process, which occurs in the matrix, is the electron transport. What do you think? What do you think it is? There you go. Now, let's start with the molecules now. The starting molecule, which has 6 carbons, number 4. No brainer, right? Glucose. Glycolysis literally means glucose splitting, so we split it into 2 molecules at number 5, pyruvate.
Pyruvate is
Yeah. Oh, well, the first thing that you get when you split glucose is something called P-gal. It's a different type of sugar. There's actually multiple different things, but at the very end, what you end up is pyruvate. You don't need to know the details. It's just regular biology. Right? You make 2 ATP here. Now that 1 molecule of pyruvate comes into the mitochondria, we lose one of the carbons as carbon dioxide gas. Not right now.
Yes.
Number 6 is acetyl coenzyme A. That's what goes into the Krebs cycle. Acetyl coenzyme A. Now 7 is the gas that's released from the Krebs cycle. Gee, I wonder what that is.
The Krebs cycle.
What do we breathe out? Carbon dioxide. 8 and 9, in whatever order, are the 2 electron carriers that are loaded up. Some are loaded up in the Krebs cycle, some are loaded up here, some are actually loaded up right here. I'm missing an arrow. We're actually loading up some right here too. 8 is NADH, and 9 is FADH2. Chris, can I help you?
He wants to pick up the camera. Your selection though?
No, you can take it.
Take it, man. Just take it, yeah. Chris, please.
10 is a gas that comes in and accepts the electrons. That's going to be oxygen.